Microcrystalline cellulose extracted from native plants as an excipient for solid dosage formulations in drug delivery. In this presentation, our goal is to offer you a quick summary of the research and conclusion. Please follow the links to review the research in further details. Thanks to all the authors and publishers for making this research public, thereby advancing science and innovation. Among all pharmaceutical dosage products, oral solids are the most commonly used by consumers to treat diseases or relieve pain. These forms are obtained by different methods that involve mixing the active ingredient with adequate excipients to obtain a solid product such as a capsule, tablet, or chewable tablet. Excipients represent the complement of the active principle in any pharmaceutical form. Their function is to provide stability, protection, and to ensure absorption of the drug and acceptability by patients. Cellulose is a conventional excipient in many pharmaceutical solid dosage products. Most of the sources used to extract microcrystalline cellulose come from cotton or wood, which are expensive and in high demand from other industries. As plants are considered the main source of excipient production, the researchers have taken advantage of the biodiversity in Ecuador to evaluate microcrystalline cellulose extracted from bojo, a native plant, as an excipient from solid dosage formulation. The method of choice for tablet manufacturing was direct compression, since it is a conventional fabrication method in the pharmaceutical industry. First, the researchers performed scanning electron microscopy, or SEM, Fourier transform infrared, or FTIR, First, the researchers performed scanning electron microscopy, or SEM, Fourier transform infrared, or FTIR spectroscopy, and X-ray diffraction, XRD, in order to compare the structures and characteristics of the extracted cellulose with two reference commercial cellulose materials. Second, they performed quality tests to evaluate the use of the isolate as an excipient, including fluidity, hardness, friability, and disintegration. Compared with commercial and microcrystalline cellulose, the extracted cellulose from native plants showed comparable characteristics as a consequently and potential excipient that could be used in the pharmaceutical industry. Last, dissolution test was performed, which concluded that all tablets have a sort of release time of active principle. To summarize, according to the characterization techniques, bojo cellulose shows a similar structure, shape, and size as compared to commercial microcrystalline cellulose. Although the BC crystallinity index was the lowest of the three samples, it seems to have a good crystalline content. Powders of the extracted cellulose also showed good characteristics and acceptable values of moisture content and fluidity, which positions them in a good potential to be used as an excipient. The quality tests on the final tablets showed that tablets composed of bojo cellulose excipient could be used without any problem of fracture, abrasion, or lack of disintegration. For future investigations, it should be considered to test bojo cellulose for use as an excipient for immediate release tablets, or to use cellulose extracted from other native plants with a higher crystallinity index to explore the diversity these would give as excipients in pharmaceutical applications.